Hi folks, a student requested some additional clarification on this table, which I'm happy to provide. So this is on the self-assessment worksheet, and Part C is basically taking a quote another look at the cell cycle and mitosis, and it's a very much a synthesis level thinking question. So let's go through it. This got deleted, I'm sorry. It says assume the amount of DNA, not the number of chromosomes in a somatic cell prior to DNA duplication is 1x. So what this means is we're looking at before the S phase of interphase. That means prior to DNA duplication, because S phase of interphase is when the DNA duplicates, which means after DNA duplication, the amount of DNA would be represented as 2x. Okay, so let's take a look at the first row here and see if we can make sense of it. So it says how much DNA, 1x, 2x, etc., is present per cell at the beginning of the process. So at the beginning of interphase, before mitosis, this statement said that that amount is 1x. After interphase of mitosis, that means S phase has already been completed, that amount of DNA would be 2x. It says place, well actually let's just work our way through this one, then we'll come back to the last two columns. So in prophase, how much DNA is present? Well, prophase it's 2x because it was 2x at the end of interphase, so that hasn't changed going into prophase. At the end of prophase, it's still 2x. At the beginning of metaphase, it's still 2x. At the end of metaphase, it's still 2x. At the beginning of anaphase, it's still 2x. Even though the chromosomes have separated, the cell is still intact. So at the end of anaphase, it's still 2x. At the beginning of telophase and cytokinesis, and I'm putting these two together because it just avoids adding an extra step on, it's 2x. But after the end of cytokinesis, right? There's telophase and then cytokinesis. At the end of cytokinesis, the cell has formally separated. So now that means the amount of DNA has been cut in half. So 1/2 of 2x is equal to 1x. So I hope that helps. The answers are provided, but I hope that verbal explanation of what's going on is helpful to you. Now in this column it says place this symbol in the box for each stage during which DNA is replicated. Is DNA replicated in prophase, metaphase, anaphase, or telophase? No, or cytokinesis? No, it's not. So the only time it's replicated is during interphase, what we just mentioned. Next column, place that symbol in the box for each stage during which sister chromatids are paired. Now, in the answer key, I put a little note here. After S phase of interphase, the sister chromatids are paired, but you can't see them. So this would technically be checked. You can definitely see the sister chromatids paired in prophase. You can see them paired in metaphase. However, in anaphase, that's when the sister chromatids separate. So you will no longer see them attached in this phase or in this phase. So the symbol would appear here, here, and here. So I hope this little verbal explanation of the table is helpful. And if you have any other questions, please post them in the forums. And thank you to the student who made the suggestion about the video.